So, dear students, continuing from the previous topic, after completion of the structure of ovule, now we are just going to functioning of the ovule. The functioning of ovule means to say ki how the ovule is working for that, what is the objective of ovule. So as earlier only we have discussed, the function of ovule is to form the megaspore, that means the formation of the egg. So that is what we are just going to start today. So I'm just going to discuss two topics today, that is what first one is megasporogenesis, how the megaspore is formed, and secondly how these megaspores are going to develop the female gametophyte or development of female gametophyte. So let us start with the megasporogenesis. Megasporogenesis is the formation of megaspore from megaspore mother cell in the nucellus. This is very much a straightforward answer. I don't think you will have any kind of the problem there, as mega spore means the mega spore, big spore, that means earlier only I just explained you, mega word is always used for the females and the micro word is always used for the male. So micro word we left behind, we are just moving on to the mega word. So mega spore means what? The egg. Genesis, formation. So formation of mega spore or formation of egg cell is called as megasporogenesis. And the important point is here that where the process is completed, the process gets completed there in the nucellus. Nucellus, if you have remember, nucellus is the major part of the ovule. Ovule is developed from the nucellus, and nucellus was originated from placenta inside the ovary only. So let us see how the process is going to complete, how the megasporogenesis completes, how the megaspore is being formed. So I'm just going to explain the process here. But remember, the process of megasporogenesis and microsporogenesis is almost similar. Only the difference is of that there in microsporogenesis we have studied firstly the development of microsporidium. Here we are not going to discuss like that, because we have completed the structure of nucellus, and nucellus is itself responsible for the formation of megaspore. So listen, the process, what is the process? I'm just going to simplify the complete process. The entire process I have just simplified so that you can understand the process easily and you will not have any kind of the problem after completion of the topic. So what happened, if you have remember there in the microsporogenesis, I'm just going to make a revision of that also. There are also some of the cells, some of the meristematic cell there in the region of hypoderm will start work as the archisporial cell. They will undergo in the process, they had undergone in the process of division, and due to that two different type of the cells were formed. That time the cell is parietal layer, and second one is sporogenous tissue. Parietal layer form the wall layer, tapetum, endothecium, epidermis, and the sporogenous tissue form the microspore mother cell, and finally they are just going to convert in form of the microspore. If you have clear all those things, there is nothing left in the megasporogenesis. Only you have to replace the terms, as I'm just going to do. What happened there in the process? One of the hypodermal cell in the nucellus. So the point or the site where the process is getting complete is nucellus. Nucellus is the structure where the process is getting complete. So any of the cell there in the nucellus, what cell should be hypodermal in the hypodermal cell in the nucellus? If you have remember, hypodermal cell we have used there in the microsporogenesis also. So hypodermal cell there in case of megasporogenesis also, any of the hypodermal cell in nucellus will start increasing in the size by absorbing the nutrient and material from the peripheral cell, and when it enlarges in the size, it differentiates from the other cell. Are you getting my point? What it does, it start enlarging by imbibing the nutrient from the peripheral cell, and as soon as it enlarges in the size, it get differentiated from the neighboring cell, and when it enlarges, it is going to convert in form of a cell that is called as archisporial cell. If you have remember, there also we have just gone through, any of the cell lying in the hypodermal region is going to convert in form of the archisporial cell, and here also we are just seeing the archisporial cell is formed. So till now what is there? Any of the cell which is present there in the nucellus region, hypodermal region of the nucellus, differentiate and enlarge in size and will start working as the archisporial cell. Similar to the microsporogenesis, that will undergo in the process of mitosis, and due to the mitosis two types of the cell will form. The first cell will form the peripheral layer, that is called as primary parietal layer, and the second one is going to form the primary sporogenous cell. Are you getting my point? First type of the cell is called as primary parietal layer, will form the primary parietal layer or primary parietal cell, and the second one is called as primary sporogenous cell. Primary parietal layer undergo in the one or two mitosis and will form, somewhere it is written, they will form the wall of the embryo sac, but our target is that primary sporogenous cells. So what happens there in the primary sporogenous cell? Primary sporogenous cell directly start, this is very important point, this is the difference from the microsporogenesis, because in microsporogenesis we have seen so many processes, the plasmodesmata, callus formation, then they are just being started working as the microspore mother cell. But here the the primary sporogenous cell directly starts working as the megaspore mother cell, MMC, the megaspore mother cell, MMC. Is it clear? Here the primary sporogenous cells directly start working as the megaspore mother cell, MMC. But remember, the crucial point of the question or crucial point is, which cell will start working as the megaspore cell? The cell which is lying toward the micropylar end. Remember, yes, in the previous lecture only we have found that the nucellus is having two regions, one is cellulose and one is the micropylar end. So the sporogenous cell which lying toward the micropylar end will start working as the megaspore mother cell, means it will lead the entire process of development of the megaspore. Is it clear till now? So we are moving ahead. As soon as this megaspore mother cell is produced, this megaspore mother cell will undergo in the process of meiosis first and second and will form 4 haploid megaspores. Is it clear? Till now the cells are diploid. Primary parietal cell diploid, primary sporogenous cell is diploid, the megaspore mother cell is diploid, but after meiosis it is getting converted in form of haploid cell, and this is going to form the 4 haploid megaspore. And all these megaspores are being arranged in form of the linear tetrad. I will explain with the help of diagram also. All these cells are being arranged, all these 4 haploid megaspores are being arranged in form of the linear tetrad. Is it clear to you all? Remember, we have seen the different type of arrangement of the 4 microspores, somewhere tetrahedral, somewhere isobilateral, somewhere decussate, somewhere linear, but here all the megaspores are arranged in the linear tetrad only. After a very important point I am just going to say, out of these 4 linearly arranged microspores, the out of linearly arranged these 4 megaspores, 3 are getting degenerates. Remember, out of these 4, the 3 gets degenerate. And which cells are getting degenerate? Which megaspores are getting degenerate? The megaspores which are lying toward the micropylar end, somewhere the questions may be there ki why these megaspores are being died or why these megaspores are being degenerated. The reason behind that, the micropylar region is not having enough amount of the nutrients, so till the time nutrients are available they are just getting developed, but after the consumption of the nutrients they are just getting degenerated. Only one megaspore remain functional, and which megaspore remain functional? Just take care, this is the megaspore present in the chalazal end. So till now it is clear, the 4 megaspore which are haploid in nature formed after the megasporogenesis, they are arranged in a linear arrangement, linear tetrad. Out of these, 3 are just getting degenerated. Which one is getting degenerated? Which are lying there on the micropylar end, and only one is remain functional, that is called as that is lying toward the chalazal end. So till now it is clear. So what is the difference there in microsporogenesis and megasporogenesis? In case of microsporogenesis, one microspore mother cell is going to give rise to 4 microspores, but here in case of megasporogenesis, one megaspore mother cell is going to give rise only one megaspore. Are you getting my point? What is the difference? In microsporogenesis, one microspore mother cell gives rise four haploid microspore, but here in case of megasporogenesis, one megaspore mother cell is going to give rise only one megaspore, and this functional megaspore which is lying on the chelagial end is taking the responsibility to develop the female gametophyte. Are you getting my point? That's the functional megaspore which is lying on the chelagial end is now taking the responsibility to produce the female gametophyte. Is it clear to you all? So now once again I'm just revising in very straightforward way. The nucellus region, in the nucellus any of the hypodermal cell which is diploid in nature will enlarge, differentiate, will start working as the archisporial cell. Two type of the cell will produce from the mitosis. One is called as primary parietal cell, will form the wall layer after the mitosis, somewhere it is written, otherwise this is not being included anywhere. And then next one is called as primary sporogenous cell. So primary sporogenous cell will lying toward the micropylar end. So the micropylar and the primary sporogenous cell which is lying toward the micropylar end will start working directly as the megaspore mother cell, will undergo in the process of meiosis, will form four haploid megaspore which are arranged in the linear tetrad, and finally out of these four megaspore, three which are toward the micropyle are getting degenerated, only one remain functional which is lying toward the chelagial. And why it is functional? Because this is getting enough amount of the nutrients, since the chelagial region is made of the parenchymatous cell which have the great amount of the stored material therein. And finally this functional megaspore took the responsibility for the development of the female gametophyte or the embryo sac. Remember the difference between that. In case of microsporogenesis, one microspore mother cell will give four microspore, while in case of megasporogenesis, one megaspore mother cell will give rise only one megaspore. Okay, so continue. Just try to understand with the help of diagram. Here in case of this, the nucellus is there. In the nucellus, the complete nucellus is guarded with, or the complete nucellus is filled with the parenchymatous cell, enough amount of the nutrient. Any of the cell, this is epidermal region, any of the cell from the hypodermal region will start increasing in the size and will start working as the archisporial cell. Archisporial cell will start division. Two cells will be formed. One is called as primary sporogenous cell and second one is called as primary parietal cell or primary parietal layer. Next one, what happens? The primary sporogenous cell will start working directly as the megaspore mother cell, will undergo in the process of meiosis, will form four haploid megaspores 1, 2, 3 and 4. 1, 2, 3 and 4. They arrange linearly. There is no different pattern of arrangement as we have observed there in the microsporogenesis. Out of these four linearly arranged tetrad, or out of these four linearly arranged megaspore, three toward the micropylar and getting degenerated, only one remain functional which is lying toward the chelagial end, and that is going to develop, or that is going to take the responsibility of formation of the female gametophyte. So now we are moving toward how the female gametophyte is getting developed. So now we are continuing with the development of female gametophyte. The female gametophyte is also called as the embryo sac. Very important question we are just going to discuss as per NEET and CBS exam both. The development of female gametophyte or embryo sac. Gametophyte is also known as embryo sac. Remember the same question is there. And the process of development of female gametophyte is also called as megagametogenesis, because megaspore is produced. Now this megaspore has to produce the female gamete, that is egg. Egg is not produced till now, so now the production of egg will also takes place. So that process is called as megagametogenesis. And as we have received the cell that is called as megaspore, and previously I just told you the megaspore is taking the responsibility to continue the process of development of female gametophyte. So the megaspore is the first cell of embryo sac. First cell of embryo sac means this is the first cell which took the responsibility to develop the complete embryo sac. And one more important thing, that is I am just making you clear, megaspore is highly reduced. Highly reduced means this is not able to develop its own nutritional resources, and for its own nutrition it has to depend on the sporophyte. Sporophyte means nucellus. So megaspore is highly reduced, cannot generate its own nutrition. For getting nutrition, for absorbing nutrition, it has to depend on the sporophyte, that is nucellus. So how the megaspore is going to convert in form of megagamete, and how this is going to develop the female gametophyte or embryo sacs. So we are just going to see the process. The three ways are there to explain there are three ways in different type of the plant families that has been studied till now. The first way is called as monosporic type or polygonum type. What is the way? First one is called as monosporic type or polygonum type. Monosporic type or polygonum type is the most common mode of female gametophyte development. This is the most common mode of female gametophyte development, which is occurs are most common in all the angiosperm. And till now we have continuously taking the example of capsula. So once again, the capsula is also having the monosporic mode of embryo sac development. Why this is called as monosporic? Because the complete embryo sac is developed from the single megaspore. Why this is called as monosporic type? Because the complete embryo sac or female gametophyte developed from the single megaspore. I'm just going to explain with the help of this diagram only. Till now what we have seen, the megaspore is there, it is present here, it is being produced, and which is remain functional in the chalazial end only. It is haploid in nature. Now what it does, it absorbs the nutrition, enlarges in the size. What it does, it absorbs the nutrition, enlarges in the size, and after getting the enough amount of nutrient present here, it will undergo in the mitosis. The first mitosis will be there, and you can understand very well, after the mitosis what will happen, one nucleus will divide into the two nucleus. One nucleus will divide into two nucleus. Now after the first mitosis completed, these two nucleus will enter into the second mitosis. Now the 2 to 4 nucleus will be produced. Finally the third mitosis will occur, and due to that third mitosis what will happen, you can understand very well, 4 to 8 nucleus will be produced. Are you getting my point? First, what happened? The single megaspore enlarges in the size by increasing the by absorbing the nutrient. Now it will undergo in the process of mitosis. First 2 nucleus, then mitosis second 4 nucleus, then mitosis third 8 nucleus. Now these nucleuses are being arranged properly in a proper arrangement pattern. How they are just going to arrange? After the division, after the third mitotic division, 4 nuclei are getting arranged on chalazial end, and 4 are getting arranged on the micropylar end. 4 are getting arranged on the chalazial end, and 4 are getting arranged on the micropylar end. But one from each side, means one from the chalazial end and one from the micropylar end, moves toward the central region. Are you getting my point? One from the micropylar end and one from the chalazial end moves toward the central region, and they will form the diploid structure called as the polar nuclei. Are you getting my point? Is it clear to you all? So now I am just going to make some more thing clear about these nuclei. So nuclei which is present there on the micropylar end and the chalazial end surround with the cytoplasm. That is very usual process. Whatever the nuclei is now present here, these nuclei will get surrounded with the cytoplasm here, and after surrounding with the dense cytoplasm, they will undergo in the process of cytokinesis, and after cytokinesis they all will just get converted in form of the mature cell. Are you getting my point? What will happen? These nuclei will get surrounded with the cytoplasm, and they will undergo in the process of cytokinesis, and finally they are producing in form of a mature cell. Okay, so what will happen this arrangement, and what is the fate of all these cells? Now just understand, the cells which are arranged at the micropylar end, they are just going to form the egg apparatus. the 3 cells which are arranged at the micropylar, and they are just going to form the egg apparatus. Remember, very important, all these cells are haploid in nature because no diploid cells are there. All the cells are produced from the megaspore, and megaspore is haploid, and the megaspore has undergone in the process of mitosis only. So all the cells are produced from the megaspore is haploid in nature. So in the egg apparatus, the egg apparatus is lying on the micropylar end. So 3 cells are present at the micropylar end. They will produce the egg apparatus. So egg apparatus is having 3 cells. So what is there? The cell which enlarges in the size and appears differentially from the other cells is work as the egg cell. My dear child, my dear student, the target of the topic is to produce the egg cell. Now the egg cell is produced. The cells which are present toward the micropylar and egg apparatus, they will form out of these 3 cells. One cell will enlarge in the size, and that enlarged cell will be considered as the egg cell, and rest of the 2 cells are called as synergid cells. So is it clear to you all? In the egg apparatus, 3 cells are there, 1 egg cell and 2 synergid cells are present. I think it is clear to you all. Now just see the 3 cells which are present on the opposite to the micropylar end, means onto the chalazial end, they are now called as antipodal cells. They are now called as antipodal cells. So is it clear? The 3 cells present there in the micropylar end are going to make the egg apparatus. Egg apparatus is made of 1 egg cell and 2 synergid cells. These all cells are haploid in nature. The cells which are present on the chalazial end are called as antipodal cell. They also are the haploid in nature. But the cells which are produced there in the middle region, because they are made from the fusion of 2 nuclei, 1 nuclei from the chalazial end and 1 nuclei from the micropylar end. So they are diploid in nature. They are diploid in nature. They are fused to form the secondary nucleus or the polar nucleus. Are you getting my point? So this is called as polar nuclear or the secondary nucleus. So this is only the diploid structure here. Now just take care. This kind of development is called as monosporic or polygonum. Why polygonum? Because first time this is reported in the polygonum only, most common in the angiosperm, even though capsula, what we are just studying again and again. And very important point I'm just going to discuss, I'm just going to say, this type of embryo sac is called as 7-celled 8-nucleated embryo sac. This is called as 7-celled 8-nucleated embryo sac. My dear child, just take care this line. This line is very important because many of the time in the CBSE examination this question is asked. Just explain the structure of 7-celled 8-nucleated embryo sac, or they are asking why the embryo sac is referred as 7-celled 8-nucleated embryo sac. So I think it is clear to you all why this is called as 7-celled 8-nucleated embryo sac. Dear students, in monosporic embryo sac only I'm just saying sharing an exception also. The exception, monosporic, it is monosporic since only 1 megaspore is taking the responsibility of development of embryo sac. What is the exceptional case? The exceptional case in monosporic embryo sac is called as Oinothera type. Oinothera type means the example is Oinothera itself. It is reported there in the Oinothera lamarckiana. So what is the specific about that? This is not a 8-nucleated. This is not a 8-nucleated embryo sac. Instead, this is only a 4-nucleated embryo sac. Are you getting my point? What is the exceptional case in monosporic embryo sac? Normally the embryo sac are 7-celled 8-nucleated, but the Oinothera type of embryo sac is only 4-nucleated embryo sac. How this comes to get that situation, how only 4 nucleus are present? So remember, in case of Oinothera type, all the antipodal cells are absent, means 3 nucleus are now not present here in case of that, and the central cell is having only 1 nucleus. So 3 nucleus present in the micropylar end, means in the egg apparatus, and only 1 is present there in the central cell. So is it clear now? 3 plus 1, 4 nucleus is there. And very important point this time I'm going to discuss, that we'll discuss later also, but here because this is very specific case, very crucial case, that is why I'm just discussing here. if such kind of the embryo sac is going to participate in the fertilization, so they will produce only diploid endosperm. The endosperm will be diploid, not the triploid. Okay, so till now it is clear to you all, I think. Now I am just moving on to the next one, that is bisporic type or allium type. The first one is monosporic. Monosporic, only one megaspore is taking part. First mitosis, second mitosis, third mitosis, and then the arrangement you have seen. Bisporic type or alien type. Allium type means what? The example is Allium cepa, Allium sativum. So that is being reported in allium only. That is why this is called as bisporic type. Why this is bisporic? As usual from the previous one, bisporic means when the embryo sac develop from the two megaspores. Are you getting my point? In the previous one, only one megaspore is taking the responsibility because three megaspores get degenerated, and here in that case only two megaspores get degenerated and two remains functional, so that all the two megaspores are taking responsibility of development of female gametophyte. So what will happen? Very usual cases, two nucleus, they will undergo in the process of mitosis. The first mitosis 4 cells, second mitosis 8 cells, they will arrange like that. So any problem here? Now the third one is tetrasporic or adoxa type, reported in adoxa. That is why this one. What is specific? All four megaspore remain functional and they are participating in the process of development of female gametophyte. All four megaspore remain functional and they are participating in development of the female gametophyte. Is it clear to you all? So now we have completed all the things here regarding the development of female gametophyte. Now the female gametophyte is ready. The students are getting confused about the division. Remember my dear child, whenever the NEET examination is there, so question may be asked the involvement of number of mitotic division. Like if monosporic type of embryo sac development is there, so you have seen the 3 mitotic division is required. If bisporic type is there, then 2 mitotic division will be required, and if tetrasporic type is there, then the 1 mitotic division is enough there. So now next one is structure of mature embryo sac. I am sharing this topic separately, but we have completed the topic detail. Right now we have completed the topic detail. Sometimes this happens, they are just asking the question, describe the structure of a 7-celled 8-nucleated embryo sac, or describe the structure of a mature female gametophyte. So this is what we have did till now. The chalazial end, micropylar end. The chalazial end is having 3 antipodal cell. All the cells are haploid in nature. Micropylar end is having the egg apparatus. The egg apparatus is having a centrally located large cell that is called as egg, and 2 supportive cells that is called as the synergid cells. Okay, and 2 central cells are there, 2 central cells out of these 4, one from here and one from here comes together and they will fuse to form a diploid cell. The polar cells are diploid in nature, antipodal cells haploid in nature, and micropylar cells haploid in nature. Only the polar cells are the diploid in nature. One new point is there, one additional point is there. The micropylar and the synergid cells are lying there, and synergid cells are having some specific point of projection over there. That projections are called as the filiform apparatus. The projections are present on the surface of synergid cells. Remember, the filiform apparatus is present on the surface of synergid cells. They are actually the outgrowth of the outer wall of the synergid cells. They produce from the outer wall of the synergid cell. This is very important in the process of fertilization because it maintains the viability of the egg by absorbing the nutrition from the nucellus cell. What it does, it helps in absorption of the nutrient from the nucellus. That is why it is helping in the maintenance of the viability of the egg cell. Secondly, this is very important point, it is going to release certain chemicals. These certain chemicals are going to attract the pollen tube toward them, means they are guiding the entry of pollen tube inside the female gametophyte for the purpose of fertilization. What it does, first of all, it absorbs the nutrition from the supportive cell from the nucellus so that it could maintain the mega gamete more viable, egg cell more viable. And secondly, it releases certain type of the chemicals which attract the pollen tube towards them. And finally, guide the pollen tube to enter into the female gametophyte for seeking the successful fertilization. So this is clear to you all about that, why this is. So 2-3 questions are there. The first question, development, or how many type of the development is there. So how many type of development? 3 basic type of development: monosporic, bisporic and tetrasporic. And why this is referred as 7-celled 8 nucleated, I just told you. 8 nucleuses are formed by the mitotic division, and these nucleuses are being surrounded with the cytoplasm, undergoes in the process of cytokinesis. So 7 cells are being formed. How the 7 cell? Because 3 are lying in the chelagel and 3 are lying in the micropylar end, and 1 is lying in the central region, that is called as central cell or the polar cell. So this is 7 celled, but the 8 nucleuses are present. 7 cells, 6 nucleuses, 3 plus 3 there on the micropylar end and the chelagel end, and 2 nucleuses lying on the polar nuclei or in the central region. So this is all about the structure of mature embryo sac, development of female gametophyte. And from the beginning only, if I'm just going to revise the entire topic, so if you have remember, we have started with the development of the megasporogenesis or formation of megasporogenesis. So this is similar to the microsporogenesis. What we have discussed today, any of the cell in the nucellus region that is lying on the hypodermal region will enlarge in the size and will start working as the archisporial cell, will undergo in the process of mitotic division. Due to the mitotic division, it will produce 2 type of the cell: primary sporogenous cell and primary parietal cell. Primary parietal cell will undergo in the 1 or 2 mitosis. Parietal cell, after parietal cell, the primary sporogenous cell will directly act as the megaspore mother cell, and where it locates? It locates toward the micropylar end. The megaspore mother cell will locate it toward the micropylar end. That will undergo in the process of meiosis, will produce 4 haploid megaspore. They all will arrange in a linear tetrad. In microsporogenesis, microspore arranged differently, somewhere isobilaterally, somewhere linear, somewhere T-shaped, somewhere decussate. That is what we have discussed. So here one difference is there. Now what happens? Out of these linearly arranged megaspore, 3 toward the micropylar end get degenerated. Only 1 remain functional, which is lying on the chelagel end. This is very much crucial point. The megaspore which is present toward the chelagel end is called remains functional, and will take care or will take over the complete responsibility of development of the female gametophyte. So I think now today's topic is very much clear to you all. So for the further studies, we'll move on to the next topic. Till here we have completed the process of development of female gametophyte, process of development of male gametophyte, process of development of male gamete, process of development of female gamete. Now the structures means what? The male gametes are ready to enter into the female gamete, and female gamete is ready to get fertilized and will start the process of seed formation or embryonic development. But before that, we have to understand the journey of male gamete or journey of pollen grains to the female gametophyte, how it occurs. So from tomorrow onward, from the next in the next lecture, I will just start the process of pollination, how the pollination occurs, how the pollen grains are shifting from one plant to another plant. Okay, so till the time, thank you.